This protocol provides a unique tool for the generation and isolation of chlamydia with altered developmental profiles, ultimately allowing for the identification of the genes that regulate the developmental cycle. The main advantages of this method are the ability to track chlamydial cell type development in real time and the ability to isolate chlamydia with altered developmental programs.
To mutagenize a Chlamydia trachomatis reporter, thaw a chlamydial stock containing approximately 3 times 10 to the 7th elementary bodies transformed with the reporter plasmid of interest, and pellet the thawed stock by centrifugation. Use sonication at 10% power for 10 seconds to resuspend the elementary bodies in 100 microliters of axenic metabolism buffer, and split the resulting elementary body suspension equally between 2 1.5 milliliter microcentrifuge tubes. Add 50 microliters of freshly prepared EMS metabolism solution into the chlamydial aliquots for mutagenesis, and 50 microliters of axenic metabolism buffer to the chlamydial aliquot only for stock mutagenesis, and incubate both samples for 20 minutes at room temperature.
EMS is a known carcinogen. Wear gloves during handling and soak all EMS-contaminated equipment and medium in 1 molar sodium hydroxide for 24 hours before disposal.
To set up a host cell culture for the imaging and isolation of the mutagenized Chlamydia trachomatis, seed 6-well glass bottom plates with 6 times 10 to the 5th Cos7 cells in 2 milliliters of complete medium per well, and seed a 24-well polystyrene plate with 10 to the 5th Cos7 cells in 1 milliliter of complete medium.